Hello, my name is Christopher Henriksen, and today we'll be going over DNA fingerprinting in crime. We have an age-old crime here, and I drew a small diagram to make it more clear. My cat's food is missing, and I know my cat did not eat it, and he's an outside cat. I know there are 4 other outside cats that are in the area, and I wanted to know who ate my cat's food. So I did whatever any reasonable person would do, is I wrangled up the 4 other outside cats, plucked one hair from each one, plucked a hair from my cat, and ran a quick DNA fingerprint to conclusively figure out who stole my cat's food. So we have 2 VNTR loci where a single DNA sample from each individual had the evidence amplified for 2 different VNTR loci and run on separate gels to create a DNA fingerprint. And we have our evidence locus, victim locus, and our suspect loci. So we can see here the evident locus actually matches up with suspect 1. The victim must be homozygous for that allele or that loci. We can see suspect 1 and suspect 2 actually are a positive match for evidence locus B, whereas suspect 3 and suspect 4 do not match. But, how do we figure out if it was suspect 1 or suspect 2? They're both positive matches. So we go on to our second VNTR loci, our second fingerprint, and we see that we have a different evidence locus A. The victim is again homozygous on this locus. We can see suspect 1 here is a positive match. Suspect 2 is not because it only shares 1. And suspect 3 is also a positive match, whereas suspect 4 is homozygous at that allele. And because suspect 1 was a positive hit on both of the VNTR loci, we can conclude that suspect 1 or cat 1 is the cat that ate my cat's food. Another interesting little fact here is that suspect 3 and 4 are likely related as they share both locus B, and suspect 4 is homozygous on locus A and suspect 3 is heterozygous. However, this does imply that they are related. So in conclusion, suspect 1, cat 1, is the cat that ate my cat's food, and we figured that out with DNA fingerprinting.